Hi, welcome back, and welcome to your final installment for the requirements in Unit 1. We'll be discussing Chapter 5 today, chemical messengers and cell signaling. The highlights for mastering concepts in Chapter 5 are highlighted here in green. Intercellular communication, which is communication between cells, the messenger molecules called ligands, and signal transduction mechanisms inside the cell or intracellular. So in this chapter, we need to consider what we learned so far in this Unit 1 material, that cells work as a team to form tissues, and tissues work together to form organs, and organs work together to create systems. There are 4 primary tissue types, which were introduced in Chapter 1. Now let's look at how cells communicate with each other, and you can think of a tissue or an organ as we go through these different descriptions. So when cells are touching or in close proximity to each other, they can use structural features that help them talk to each other. But when cells are far away from each other, they need a method of long-distance communication that happens in the form of a nervous signal or an electrical impulse, which we'll spend all of Unit 2 learning about electrical impulses. But they can also use a chemical messenger called a ligand. It's similar to how hormones work in your body to communicate with distant tissues or organs with the help of the bloodstream. Communication between cells and within cells is what we need to start with. So cells can detect what's going on around them, and they can respond in real time to cues from their neighboring cells and their environment. At this very moment, your cells are sending signals and receiving millions of different messages in the form of chemical signaling molecules. These signaling molecules are called ligands. Communication between cells that are in direct contact with each other, like we see here in Figure 5.1, Panel A, there's a conduit between these cells, and molecules can pass directly from one cell to another through what's called gap junctions. Gap junctions are made from a protein called connexin. This is a rapid form of communication since the cytoplasm of each cell is connected as one. And again, this is only happening when cells are in direct contact with each other and can form this conduit between each other. So they act as one. It's a very rapid method of cell-to-cell communication. But when cells aren't touching, then an indirect method is needed, and chemical messengers, called ligands, are released into the ECF compartments, also known as the interstitial fluid, and the target cell must display a protein receptor facing out into the ECF in order to hear the message from the other cell. So that's the case when the cells are a little farther apart and they're not directly touching each other, like what's shown here in Panel B. This is called intracellular signaling, and communication within a cell is called intracellular signaling. When you think of the Latin root on these prefixes, inter meaning between, like an interstate allows you to travel between states. Intra means inside, like an intravenous fluid or an IV. Okay. So here in Figure 5.2, this illustrates the 4 different methods of communication between cells using chemical signals or ligands. Remember, a ligand is any molecule that binds to a specific receptor protein on the target cell. Examples of ligands include paracrines, often used within an organ to coordinate surrounding tissues, shown here in Panel A. Autocrines help with self-regulation, auto meaning self, since the signal is formed by the same cell that is being targeted. Neurotransmitters here in Panel C are ligands released by neurons following an electrical impulse, and hormones in Panel D are chemical signals that are sent out over great distances, and these use the circulatory system for delivery of their signal to different targets. Much more of this on hormones will be covered in Unit 3. Specificity in a cell that's being signaled to, it can occur in a couple different ways. Ligands and receptors are highly specific for each other. A specific ligand will have a specific receptor that only binds that particular chemical ligand. Not all cells will have receptors for every ligand. So that means only the cells that have the receptor are capable of detecting and responding to the chemical signal. This gives us signaling specificity. We know that some chemicals will be water soluble and others will be hydrophobic or fat soluble. Once you determine the nature of the ligand, you'll know exactly how it will interact with its target cell. So, if it's a nonpolar ligand, will interact well with the fatty acid chains on the interior of the plasma membrane where the lipid portion of the bilayer is. This means nonpolar or hydrophobic ligands will gain access to the interior ICF compartment on their target cells by simple diffusion across the plasma membrane, so no need for a protein receptor. Steroid hormones act in the same manner. However, when you have a water-soluble polar hydrophilic ligand, then the surface proteins must bind in the ECF compartment, and now they need help to tell the inside of the cell to change or do something in regards to that signal. The binding of ligand to receptors produces a response in the target cell by what's called signal transduction. And now we need a secondary messenger. So the ligand is going to be stuck outside of the cell, and we have to communicate with the inside of the cell. Here in figure 5.4, this is a reminder of how protein synthesis occurs in the cell secreting the message. You can see the secretory vesicle helping to transport the newly made protein or peptide towards the plasma membrane for exocytosis from the cell out into the ECF or interstitial fluid compartment. Once the signal's been released and it's outside the cell, depending on the ligand's solubility, it may or may not like being in a watery environment. When there is a hydrophobicity with the ligand, it will need to be stabilized during transport with a carrier protein. The bloodstream is a watery world, but there are proteins dissolved in blood, so carrier proteins will help stabilize a hydrophobic molecule until it arrives on scene at its target. This is compared in panel A and B in figure 5.7. When the ligand arrives at the target cell, there are three types of surface receptor proteins that can be used. There are ion channels, there are enzyme-linked channels, and there are G-protein linked channels. These three different surface proteins are required when a chemical ligand is water-soluble and cannot directly access the interior of the cell. In this case, a secondary messenger is needed to communicate to the inside of the cell that the ligand is bound to the outside of the cell. When this happens, the original signal is amplified by using signal transduction mechanisms. These amplification processes can have a huge impact on target cells, resulting in activities like altered membrane permeability, stimulation of protein synthesis, activation or deactivation of different enzymes. They can induce secretory activity in the cell and even stimulate mitosis. Those are major cellular events. Here in figure 5.13, we can visualize a fast mechanism used by cells to convey a water-soluble ligand, which is signaling to the cell that something needs to happen. So that's only going to be allowed to access the outside of the cell because it's water-soluble. So we have to have a protein receptor to catch the signal in the ECF compartment. So here on the left, we read this diagram from left to right, just like we would a sentence. The receptor on the left lacks the chemical ligand, so it remains in the closed position, and charged ion particles are not allowed into the cell. But if we move over here to the right, the protein now has this triangle-shaped ligand bound specifically. They kind of fit like a lock and key to the receptor. And here, this is an ion channel receptor. So when it opens, It allows the passage of ions by diffusion across the cell membrane. This acts like a gating mechanism, and due to the established ion concentrations inside the cell, these were listed out for you in Table 4.1, we can see that sodium rushes into the cell and potassium is lost due to their natural concentration gradients. As long as the ion channel is in the open position, the cell will continue to lose potassium and take on sodium, which ultimately disrupts the electrical properties and depolarizes the cell. More on this behavior next week during Unit 2. In Figure 515, the G protein receptor is activated by a water-soluble ligand and that binds its protein receptor and must then activate a series of secondary messengers inside the cell. And it starts this by releasing one of its 3 peripheral protein subunits, given the Greek letter names alpha, beta, and gamma. These are peripheral proteins that kind of hang out on the inside surface of the cell, whereas the receptor itself is an integral membrane protein. So it touches the inside of the cell and extends out into the ECF. And you can see this labeled as step number 1. Okay, so the triangle-shaped ligand binds, and in step 2, we have this lateral movement. Okay, so at the expense of GTP, which is a close cousin to ATP, the energy for this lateral movement of just the alpha subunit, it's going to move to the right towards a neighboring ion channel, and it activates it from the inside of the cell. And that ion channel is labeled as number 3. Again, once the ion channel is in the open position, it's like punching a hole in the cell, and the ions are going to flow in the direction of their concentration gradient. In this figure here, we see a G protein operated receptor that activates an enzyme called adenylate cyclase. Note the suffix ase always indicates a protein enzyme. Let's follow the numbered steps here. So first, the ligand binds to its receptor. The energy from GTP then activates the lateral movement of the alpha subunit. And then in step 3, when it reaches the adenylate cyclase, that enzymatic activity burns an ATP for more energy that results in a cascade of secondary messenger events inside the cell. Step 4, cyclic AMP is activated, and its job is to activate protein kinase A, which results in step 5, where intracellular proteins become phosphorylated, causing an overall cellular response. We will run into some common secondary messengers. Cyclic AMP is shown here, step 3. Inositol triphosphate, or IP3, is one we'll talk about in muscle activation in Unit 3. Diacylglycerol, or DAG, and calcium. Believe it or not, calcium ions are very strong secondary messengers and can cause massive changes in the cell. I'm going to refer you to your textbook on page 140 for more on this with calcium and diacylglycerol. We mentioned earlier in this lecture some of the major activities that can be stimulated to occur using these types of signaling mechanisms. Things like mitosis, activation of secretory pathways, protein synthesis, among others. These are important steps to become familiar with. So once you're done with the lecture notes and reviewing, why don't you go ahead and just please watch the selected video demonstrations of these principles kind of in motion. We can put some of these diagrams from our textbook into motion so you can see how there's activation and we have a cascade of events, and kind of get in your mind these numbered processes moving around and what they do inside the cell. So you'll want to watch the videos and complete Chapter 5 before sitting for the exam later this week. Thank you.